Thanks to the organisers for inviting me to this great event that I wish I could be at for longer and avoid all my teaching. So my exam was last week, so I got out here and it's been great. So since David Hoyle was already here and talked about our theory work, I'm going to talk about something completely different, which is joint work with Neil Lawrence, who's now at the University of Manchester as well. So I'm going to talk about transcriptional regulation of genes, and I'm going to talk about a couple of different types of statistical modelling we've been doing for this problem. In the first part of the talk, I'm going to talk about really small-scale systems which can be described using differential equations. I'm going to talk about how we can do inference over unobserved continuous time quantities in these types of system. So the system is deterministic, and but probabilistic inference is still useful because these continuous time unobserved quantities are not fully specified by the data. And in the second part, I'm going to talk about larger scale models for the entire genome set of genes. So different types of approaches are necessary there because of the scale of the problem. So the idea with gene expression is that there's a promoter region in front of a gene in the DNA, and some molecular machinery has to find the start of the gene in order to start making the gene. And the gene is first transcribed into messenger RNA and then translated into a protein. And there are a number of things that help this process along. And one of those is a transcription factor protein. So there's a large class of proteins, very diverse class of proteins called transcription factors. They work in many different ways. So this is really a cartoon. There's lots of different ways that these things can work. But one thing that can happen is that a activator transcription factor can bind energetically to some sticky part of the DNA and help the process of this machinery finding the start of the gene along and increase the rate at which the gene is produced, leading to more proteins in the end. But the point for this talk is that it's easier to measure the mRNA than it is to measure the proteins. And you can also get repressor transcription factors. So something could bind which actually blocks the activity of this RNA polymerase and slows down the rate at which expression happens. This thing is binding, unbinding, binding, unbinding. So this is, on average, it could be blocking something completely, but on average, over time, if you look at this process, then you'll see that the rate of transcription is reduced. It's not necessarily stopped completely. So you can model this type of process using ordinary differential equations. So here's a really simple ordinary differential equation which was used in a similar type of statistical inference by Barenko et al. So the idea here is that X is a gene's messenger RNA. It's a concentration of messenger RNA at a particular time. It's produced according to some sort of baseline production rate, and then a rate which is influenced by this transcription factor's concentration. So the transcription factor is a protein, and when it's in the nucleus, it helps this transcription along. And then it decays with a constant here which is indicative of how stable the mRNA is. So if the mRNA is very stable, then it decays very slowly. If the mRNA is very unstable, then it decays very rapidly. And when you look at mRNAs in the cell, the stability of the mRNAs is very, very different. Some of them are incredibly unstable, just decay away almost immediately. Some of them hang around in the cell for a very long time, and their rate actually only goes down because of the cell doubling process. So there's a great variety in this constant here. And the application I'm going to use to motivate this type of modeling is identifying co-regulated genes. And that's actually what this paper by Barenko was about. So they were using this type of model to, yeah?
How do you take into account the different scales for each transcription factor? So some of them might have a large amount and some of them might have a small amount.
So large amount of?
So each transcription factor has a different sort of variability for itself relative to other ones.
Oh, right, okay, but this is for just one transcription factor at the moment. Later on we'll look at multiple. So the problem, the statistical problem here is how do we fit this model to data when the transcription factor is not observed? Okay, so this transcription factor may be regulated by a signaling pathway, for instance. So a hormone or something stimulates the cell, a message is passed down a signaling pathway, phosphorylates the protein, and it becomes active. That whole process, as Mark was talking about the other day, is quite difficult to measure or to have data about. So it's quite hard to know what F is here. So what we want to do is, but it's quite easy to measure X because we can use microarrays. So we have measurements of X, but we don't have any measurements of F. So F is unobserved, so we're going to treat that as a latent variable. So this is an important, or I'd argue this is a good approach because you can look at genes which are regulated by the same transcription factor, and you can look at time series of their expression, and they can look very different. Okay, so these two genes are both being regulated by the ALK1 transcription factor after stimulation with EGF. And this gene responds very rapidly and then decays away to a low level, whereas this gene also responds rapidly but then hangs around for quite a long time. And it turns out that this gene is much more stable. The mRNA stays in the cell even after the stimulus has gone away. So clustering can't be relied on to identify these co-regulated genes because their profiles are quite different. So if you just represent these as vectors and do some clustering, then these are going to end up in different clusters. So I'd argue that a model-based approach is more appropriate to solve this problem. So that's why we need this ODE type model, because that captures the fact that we can model the decays as being different. And one can also have nonlinear models. So for instance, a Michaelis-Menten type kinetics captures the fact that the response of the mRNA might saturate at some point. So for very large F, this function here will go to a constant. So that kind of just captures this sort of saturation. And one could have some sort of repression, and repression is sort of inherently nonlinear. So as this transcription factor concentration goes up, this quantity becomes smaller, leading to slower production of the mRNA. So the approach that we've taken to this problem is to represent this unobserved continuous time function as a sample from a Gaussian process. So a Gaussian process is a distribution over functions, and it's a convenient distribution to use for a probabilistic inference because it allows us to do posterior inference in a sort of Bayesian framework over these continuous time functions. So a Gaussian process is determined, wrong button, is specified by some sort of mean function here and by a covariance function. And the covariance function captures properties of what samples look like drawn from this probability distribution over functions. Okay, so this captures, for instance, smoothness of the function. It captures persistence, how often the function crosses a zero line. Or one could have quite rich information in here to do with periodicity. So one can put a lot of prior knowledge into these covariance functions. But we're going to use fairly vague priors, if you like. So in most of my examples, we're using this squared exponential type covariance function. And this has a parameter L here, which is a length scale. And the length scale basically determines, on average, how many times this function crosses a zero line. So how wavy the function is. Samples from this distribution are infinitely differentiable, so these are very smooth samples. So this isn't a stochastic process. It's a random process, but not stochastic in the sense of stochastic differential equations. Another smooth process is given by this, it's called an MLP covariance function, where that's multi-layer perceptron, because it's found to be the limiting case of an MLP with an infinite number of hidden weights and particular priors over the weights. So this I'm just showing because it's a non-stationary function, whereas the previous one, these functions look the same wherever you are if you translate in time. So this is stationary because it only depends on the difference between two times. This one's non-stationary, and it actually depends on absolute time. So something happens here, and then it goes off to some kind of flat behavior down here. So those are examples of covariance functions. So if we go back to the model, we have this linear regulation model. We can solve for x. And what you see is that this is a linear operation over f. And if you have a Gaussian distribution, then if you have a sum over Gaussian variables, then you get a new Gaussian variable. And similarly with Gaussian processes, if you have any kind of linear operation on a Gaussian process, you get a new Gaussian process. So if the protein concentration comes from some Gaussian process prior distribution, then the mRNA that's regulated by that protein also comes from a Gaussian process. And the covariances can be worked out by simply doing a sort of basically multiplying x's together and taking the average. And so you can work out the covariance, and for the special case of the squared exponential covariance for f, you can analytically carry out these integrals. So you can actually get closed form solutions for the covariance of the x's and the f's. So the mRNA and the protein are all jointly a Gaussian process prior. So I haven't observed any data yet, I'm just setting up the prior. Okay. So this is the joint distribution of the protein and the mRNA data. So it's this Gaussian process. And then Bayesian Gaussian process regression allows us to condition on the data and do posterior inference over the protein concentrations. So we can work out the probability of the protein concentration given some data. But X is only observed, and with noise, at discrete times. We don't observe complete functions. We only observe things at discrete times. And the nice thing about Gaussian processes is that these computations here only require us to evaluate these covariance matrices at the data points. So we don't have to deal with the functions. That's all implicit. We can do things entirely by evaluating covariances where the data is collected. So I'll give you an example. And the first example I'll give is for a case with noiseless data. Just to show that the uncertainty in F isn't due to the noise in the data, it's due to our lack of information. Okay. So this is completely deterministic data. We just have 6 data points from 3 genes. And we have genes with different kinetic parameters. So just to remind you, this was a baseline expression rate. This was a sensitivity to the protein. And this is a decay rate for these 3 genes. So I'm going to assume that I know those parameters. In general, I wouldn't, but just for the example, I'm going to assume I do and see what happens. So the solid line here is the ground truth. Okay? So this is just some artificial data that's being cooked up. And the solid dots here are the data points. So there's 6 data points measured at different times for these 3 different genes. And the top gene here has a very stable mRNA. So it has a very slow decay rate. And that means it basically integrates the signal from the protein. Okay? But the bottom mRNA here has a very high decay rate. And that means that it basically mirrors the protein's profile because it can respond very rapidly. So if the decay rate tends to infinity, the mRNA just looks like the protein. It responds immediately to the protein stimulus and then just decays away. So it's proportional to the protein in that limit. So given these 6 data points and the prior knowledge that the protein was 0 at time 0, then we can use this posterior inference to get the posterior distribution over these Gaussian, over these functions. And the dashed lines here are samples from the posterior distribution. So what you can see is that the data constrains the function completely, so it has to go through the data points. But between the data points, there's a little bit of variability. And the further away you get from the data, the more variability you get. Then after the data here, there's no information to constrain the function, and it just diverges towards the value given by the prior. Okay? So in the figures I give, I'm going to summarize those kind of posteriors using the marginals. So this shading here is the marginal of the posterior, and the black line here is the mean for my posterior inference. But you have to remember that the marginal isn't that the posterior is a posterior of a function, so there's more information. You really have to sample from it to see what the things look like, okay? Because this marginal doesn't really contain the information about the time correlation in the posterior functions. You could have a stochastic function that had the same marginal. So that was inference if you knew the model parameters, but in general you don't. And part of the problem we want to solve is to infer the model parameters. So we can do that by creating a likelihood function for the model parameters and integrating out the uncertainty in the protein And that's quite nice to do because the protein's this function, so it's infinite dimensional. So maybe it would be a bad idea to do maximum likelihood on that. So you might want to integrate that out and do either maximum likelihood on these parameters or do some sort of MCMC to infer their posterior distribution. There's quite a few parameters for quite a small amount of data, so I think MCMC is probably a good approach here, actually. For the examples I'm showing, I'm just using maximum likelihood. So here's another artificial example, but this time it's with realistic parameter values from a data set, from the Barenko data set. And this time the data is noisy, and we're going to do inference from the noisy data of the parameters and the function. Okay? So the data are these solid points here, and the truth is the dashed line, and the solid line is our inference, and the shading gives us 95% credibility intervals for the posterior. And what you can see is that with this amount of data, you can pin down the function quite nicely, despite the fact that it's got this quite difficult nature to deal with because there's two stimulus here. You can see that there's a little bit of waviness at the end here, and that's probably due to the prior over these very smooth functions. So this RBF, this squared exponential prior, tends to like these very smooth functions. So that means there's a penalty in quickly tapering off and going flat. The function tends to want to be smooth here and smoothly go back to the 0 point. So perhaps there's an issue here with the prior, which is leading to some errors in the inference here. But nevertheless, it works quite well. And for this data, this amount of data, you basically infer the parameters of the model very well. So the white here is the truth and the black is the inference, and there's a very good match between the inferred parameters and the true parameters. It's quite a lot of data though, and more than perhaps typical for microarray experiment. So then we can look at some real data. So this is Barenko's data set. So they identified 5 training genes in order to build their model, which were definitely regulated by the p53 transcription factor. And so we applied this to their data. This time we have a gene-specific variance parameter as well, so that's why we're getting these differences in the credibility intervals for the different genes. What you see is that genes regulated by the same transcription factor can have quite different peak times, which is due to the fact that the decay rates are different, mainly. So something that has a slow decay rate tends to peak a bit later after the stimulus. So in terms of an application, in Manchester, we're working with a biologist who's interested in targets of particular transcription factors, and he's interested in the ALK1 transcription factor and has carried out chip-chip experiments to work out which genes this transcription factor binds to physically. And what he wants to do is integrate his chip-chip data with expression data in order to work out in specific circumstances which of those bound transcription factors is activating its targets. So he supplied us with these 5 training genes which are confident, he's confident are regulated only by this transcription factor under the conditions of the experiment. So most genes are regulated by more than one transcription factor, but in this case, he believes that these are primarily regulated by this one in a time-varying manner. So the microarray data comes from the Samatop et al. Nature Genetics paper. which remarkably, they only have one replicate of the experiment, and they still get into Nature Genetics. Which is really quite upsetting because there's something wrong with the fourth data point, but we have no replicates in order to deal with that, really. But never mind, we still get something reasonable out of the data. What you see is that the training genes all look fairly similar, except for this one, which is very stable and therefore has quite a different profile. But the fit to the model for all the genes seems reasonable, and we infer something sensible for the ALK1 transcription factor. It peaks very rapidly, then dies away in less than an hour, and then goes kind of flat. But there's quite a small amount of data to make that inference from. And so then what we did was we used that model in order to score about 1,000 genes that had come up in the ChIP-ChIP. So what these guys want to do is they want to refine their ChIP-ChIP targets because they have about 1,000 and it's too much to deal with, and really focus on the ones that they're confident about. So if we look at our top 50, then according to ChIP-ChIP, these are pretty confident, many of them, so they have a false discovery rate of less than 10% according to the ChIP-ChIP data analysis. Now, you would get a quarter just by chance, so that's, this is already a refined list. We're not just dealing with the whole genome here, because there's a lot of stuff going on. We can't really only find targets of this one transcription factor after EGF stimulation for the whole genome. But what's interesting is that some of the other 50 were confirmed as true targets by other experiments, but missed by their ChIP-ChIP experiment. Or rather not missed, but associated quite a low confidence. So that means that we can kind of refine their ChIP-ChIP data and pull out the ones which they were unconfident about and we're confident about, and that gives them a better list of top genes to go away and investigate. So that's a kind of application of this sort of modeling. And just have a look at a good and a bad fit here. This is one of the predicted target genes, and this is a gene that just doesn't fit the model at all. It peaks, but it peaks much later and can't be explained by this simple regulation model. So there's something else going on for this target gene. So that was just using a linear response model. And the linear model is very nice because if F comes from a Gaussian process distribution, then so does X, and the whole thing can be done with Gaussian process regression techniques. Very straightforward. But in practice, most responses are nonlinear. And you can still solve this differential equation. It's still a linear differential equation in X, so it's still very simple. But the relationship between X and F is now nonlinear. So if F comes from a Gaussian process prior, X no longer does, and the posterior for F is no longer a Gaussian process. So we have to resort to approximations in that case. And the two types of approximation we've used are a Laplace approximation around the map solution for the function f. So you can find that using numerical optimization. And then you can work out the posterior and the marginal likelihood with that approximation. And we've also done MCMC, where we sample this f function on quite a dense grid. And because it's on a dense grid, you have to design special Metropolis Hastings moves to deal with the fact that the variables are highly correlated. So if you have highly correlated variables, so these functions are smooth. So in the limit where the discretization goes to 0, a point between two other points is completely constrained by its two neighboring points. So you can't do Gibbs sampling for individual points, okay? Because you'll move infinitely slowly. So you have to do special types of Metropolis Hastings on these functions, and we have some tricks, but I'm not going to give them away because we're about to submit that for conference. So if we take a nonlinear activation model, so this is a nonlinear kind of activation model. This time, there's another thing I should say, is that these concentrations obviously shouldn't go negative. Gaussian processes can go negative, so that means our prior is already a bit crazy because we're allowing our transcription factor to become negative. So we're going to use e to the f as our concentration, where f comes from a Gaussian process prior. And one of the so I showed a single replicate from the Barenko data set. If we look at a different replicate, what we observe is that some of our posterior distribution does actually go negative. So this distribution is saying that there's a little bit of probability associated with the transcription factor having negative concentration, which isn't really believable. So instead, using this function for the response, we can do inference, and we get something that looks a bit more sensible, although it has a bit of a higher uncertainty, perhaps because we have this e to the f prior. And I've shown some MCMC samples. The dashed line are the MCMC credibility intervals, and the shading is the Map-Laplace approximation. So for this particular case, Map-Laplace is pretty good. Okay, so differences between here and here aren't due to differences in the approximate Bayesian scheme used. They're due to differences in the model. Which one is correct well, I guess that's a model selection issue, and perhaps some of the stuff that Mark was talking about on Tuesday would be relevant in terms of scoring these models. So we can also do repression. So again, just reminding you about what the solution to the ODE is. Now we have this transient term because previously we were assuming the transcription factor had 0 concentration before time 0, but for repression that doesn't make sense for this particular experiment that we're looking at. So we have an additional set of parameters. And this was a results for a experiment where the Lex-A protein was irradiated, so it was hit and went down to very low concentration, and then over time recovered. So you knock down this this transcription factor, which is a repressor, and then it comes back again in order to find out its targets. Okay, so you want to detect which things are repressed by this transcription factor. There were 14 training genes, and I'm just showing 3 of them just to show the difference in response that you can get for the same regulation model. And if you look at the inferred parameters that we found, these are in descending order in terms of stability of the mRNA. So this one here is very stable, and that means that basically it's integrating the response to this stimulus, and that means it's always increasing its concentration despite the fact that the transcription factor repressor is increasing. Okay? Because there's still mRNA from this bit hanging around. This one peaks and then goes down, which kind of mirrors the effect of this transcription factor. This repressor goes down and then goes up. So as it goes up, that means that it's reducing the production of the mRNA. So that's why this is then going down. This one peaks and then goes down very gradually. But what you can see is that the peak time here, this one, I guess, peaks over here somewhere. This one peaks here, this one peaks here. The peak time is related to this decay rate. And if you didn't take a model-based approach, you wouldn't be able to spot that these were co-regulated. So this is a very nice example because the responses are so different. Okay. So this Gaussian process approach, we're really pushing for these small systems. But now I'm going to move on to much higher dimensional inference problems. So in this case, we're looking at a situation where potentially hundreds of transcription factors are influencing a whole genome of genes. So potentially tens of thousands, or in yeast, 6,000 genes. And the problem here is not so much to come up with a detailed mechanistic model of how the regulation is occurring, because that's out of reach really for this scale of problem. Now we're really trying to infer what are the links in the regulatory network. How are the transcription factors affecting genes under specific experimental conditions? So for this problem, we're using much simpler linear models. So here's a factor analysis type model. So for this type of approach, we're working with a log of the expression, because if we have multiple transcription factors, then the assumption is that they have a sort of multiplicative effect in terms of the steady state expression of genes. And again, we don't know what the log concentration of the transcription factor proteins is. So we don't know C, that's unobserved, that's a latent variable. We also don't know the effect that the transcription factor has on a particular gene. Okay? So this is, as it stands, if we only had Y, probably not a very realistic problem to attempt with the amount of data that we're doing. So here we have maybe 20 experiments, 6,000 genes for yeast, maybe 200 transcription factors. So this A matrix here is 6,000 by 200, and we have 20 data points. So in order to make that problem tractable, what we do is we include a lot of prior information about the regulatory network. And this is our work, and this is somebody else who approached the same problem at the same time using the same model. It's just a bit unfortunate for us, but luckily they did Gibbs sampling and we used variational Bayes, so we were allowed to argue that they were sufficiently different. And they did a different organism, which is smaller. Yeah.
That model is not identifiable.
Well, it is with the prior. So
then you take care of that.
it's not identifiable. You're right, and there's a rotational ambiguity. So if you did this by maximum likelihood, you'd have to do some rotation of the factors, and you'd have to use some kind of I don't want to say prior, but for instance, people have done rotations to find the most sparse basis vectors, is one way of doing it. So that's true as it stands, but I haven't said what the model is yet because I haven't put any priors in. Right? So this isn't the model yet. So now it's a model because I've got some priors. So I'm going to use strong prior information. So the strongest thing I can do is just assume that the regulatory network is known. And that means that I know lots of zeros in this A matrix. Okay? And for the ones which aren't zeros, I'm going to assume a Gaussian prior. Another thing we did was we also, because we had time series data, we wanted to include the fact that these latent variables were changing smoothly in time. So we put a unit variance Gaussian process, but discrete time Gaussian process on these. Okay. See, that's not really the standard Markov chain prior. It's a bit different if you think about it. It's unit variance stationary. If you usually with a Markov chain, what you do is you have a different initial condition and proposal, but here we've constrained this to have unit variance and be stationary. It's a sort of detail, But what's good about that is that you could have one of these C's constant in time entirely, and it still give you sensible inference. Right. So this gamma variable here tells you how smoothly these C's are changing over time, and it's a number between 0 and 1. So to solve these problems, we want to do it in a Bayesian way, and I'm going to motivate that in the next slide when I look at some results. So if I write the prior in a different way, I can write this A matrix here as a binary variable times a continuous variable. The continuous variable comes from a Gaussian. The binary one is just determined by this prior probability of being at 1 or 0. So that's giving me the network. And the B's are giving me the strength of influence of the genes, of the transcription factors on the genes. So in our approach, we assume that the network itself is known, although there could be false links. And I'm going to show that you can still make inferences about links which are incorrect in the network. So there could be false links, but we don't allow the fact that links which are not there could be discovered subsequently. And we use the variational Bayes approach to approximate the posterior distribution of the A's and the transcription factor concentrations, basically assuming a factorized form for that. Sabaty-James had a smaller data set, and it was tractable to do Gibbs sampling, and they sampled over the X, A, and C, but they constrained many of the links in the regulatory network to be 0. And these constraints on the network is what makes the problem identifiable. So when you have those, it's identifiable. But in general, you're right, it's not. So we've actually implemented a Gibbs sampler that scales up the Sabaty-James model to hundreds of transcription factors. So we don't really need our variational Bayes anymore for that size of problem. I mean, if you don't, the only point in variational Bayes is if MCMC is not tractable, right? MCMC is better because if it converges, then it gives you the right posterior. So you should try MCMC first and only use variational Bayes if it fails. But one thing I'll say is that variational Bayes seems to work very well on dynamical models. So when we have our Markov process on the C's, then the variational Bayes seems to be working very nicely. And the hyperparameter estimation problem becomes quite slow in the Gibbs framework because you have an extra nested sort of Gibbs sampling move. So if we take some example problems, so if we have some artificial data, then we have some true parameters. So this is C, this is B. These are the posterior means inferred. There's a trivial identifiability problem because you can flip the sign of B and C because it's a multiplicative model, right? So, but that's kind of trivial. And variational Bayes will just focus in on one of those trivially equivalent modes. So, so really you don't care about that too much, and you can break that by just choosing the same sign as in your true model. In a real application, you'd have to use some external information in order to break that symmetry. For instance, if you knew that a transcription factor was an activator, then you could know that the B was positive. So this just shows that you are recovering the parameters, and I guess you can do this for any particular network you're interested in to see if you have identifiability. Then you can look at what happens if you have false links in your prior. So if you know the regulatory network, then you can infer these B's. If you introduce false links into the regulatory network, then you can see whether you can use your B's in order to switch off links. So you can rank the links according to how confident the model is that they have regulatory effect. As you introduce more false links into the regulatory network prior, you get poorer performance in terms of the ROC. So the ROC here, what I've done is I've ranked my links in the network according to the strength of the B regulation. I've done it in two different ways. I've just used the size of B in the posterior, or I've used the size of B divided by the posterior standard deviation. So this is kind of taking into account the variability. If you take the posterior variance into account, you get a better ROC score. As you introduce more false links into the network in your prior, your AUC goes down, and eventually, if you have 100% links, if you assume that the whole network is connected, you get no performance because it's unidentifiable. Right? So I didn't show that line in the thing, but it does go down to 0.5 for both of those in that case. Yeah. So the prior is the only thing that allows you to do the inference. And then we applied this to some real data. So everyone applies it to this Spellman yeast cell cycle data and the Lee chip data. So we did that only in order to really compare to other approaches using partial least squared and methods. And in terms of reconstructing the transcription factor profiles, some transcription factors are primarily regulated at the level of expression. So their mRNA is quite a good proxy for their protein. If you look at those ones, what you see is that the inferred transcription factor protein concentration looks like a less noisy version of the expression data for that transcription factor, and often slightly time shifted. So these are two examples. What you do find is for a particular microarray expression data set, many of the links are switched off by the model. So that's kind of what you want to know, I think, if you're a biologist. You want to know which of these links are really influential in the particular expression data that you're looking at. And this is just another example. This is the mRNA for a protein. This is the inferred protein concentration. And these are the strength of the links of genes which are regulated by this transcription factor. So many of them are around about 0 and are basically just switched off. Okay, so those are the two types of modeling we're doing on regulatory systems. But there's lots of things to do, lots of interesting stuff. So, I mean, these linear models can only describe a subset of transcription factors which behave in such a simple way. And really, transcription factors, if you want to move up to higher eukaryotes, then you're going to have to deal with nonlinearities, modularity, these kind of issues. Transcription factors work together in order to regulate genes. So these models are too simple. So Lin and Husmeier have recently done some work using mixtures of factor analysis on genes in order to capture transcription factors working together as modules. That seems like a very promising approach to extend this. Another thing they did, which I think is the correct approach, is to treat the information about the network from chip-chip data as data, not as a prior. Okay? Because you have data from chip-chip experiments. So really, you're making an observation of the regulatory network, and that's data. So your model should be X is observed with noise, and we observe the expression data which depends on X, and they should both be treated as data. So I've got a student actually looking at those two approaches, because to me it feels right to treat it as data, but then my student was saying, well, why does that give better results? So he's trying it out and seeing how that pans out. So Gibbs sampling, you know, I don't want to spend my whole life waiting for Bayesian inference to happen. So I want to investigate more sophisticated ways of doing deterministic Bayesian approximations on graph inference. So if you do naive variational Bayes where you just assume kind of a mean field assumption over the links in the regulatory network, it's rubbish. So it just doesn't work at all. So if you're doing variational approximations, you probably need to use some sort of tree based approximations or something to the graph. So we need better variational approximations to these networks. And I'm quite interested in these message passing type algorithms and seeing how they work. I'm really interested in rich priors. So I'll just do a push for our paper recently in PNAS on using phylogenetic priors for protein-protein interactions. Completely unrelated to this work. It's just an advert for a paper. But it's not completely unrelated because what we're doing there is we're using phylogenetic information to relate networks in different species. And you can do that with regulatory networks as well because we have the phylogeny and we have data from different species and we have really rich data for some species and less rich data for other species. So I'd really like to use data from one species to make inferences about other species. And you can do that with phylogenetic priors. And so that's something that I'm interested in putting into these kind of factor models. And for the ODE stuff, well, single cell data is coming now, so we want to look at stochastic differential equations, but that's a whole can of worms. So, okay, so this was joint work with Neil Lawrence, who's now at Manchester. Lots of people contributed to these works, and we got data from people at UCL, Glasgow, Lancaster, Manchester, and those are some of the papers. They're all on my website or on the project's web pages. Thanks.
How different were your results from those of Sabatier and James?
They can do our scale of systems, so we couldn't compare them on the same yeast data. They did E. coli, and they had 30 transcription factors. But my students now implemented a different type of Gibbs sampler. The problem with their Gibbs sampler was that they were sampling binary vectors, and if more than 11 transcription factors were, how should I say it? If there were more than 11 ones in one of their vectors, then when they sampled their Gaussian variables, they got a kind of combinatorial explosion in their sampler. So we're doing Gibbs sampling in a different way, which scales up, and we'll see. The models are very similar, so I expect the results wouldn't be too different.
Yes, operationally, I would think there's a lot of difficulties. If you're trying to match up expression data with network data, you have to provide some IDs
Oh yeah.
to match them.
Yeah.
I mean, maybe you're just using a small set of transcription factors and you can match them up fairly easily, but
well, there's quite a lot of. This was an issue with the human data, certainly, because the chip experiments used promoter arrays, Affymetrix promoter arrays, and the expression arrays were different, so they were the standard Affymetrix gene expression arrays. And yeah, there was an issue with mapping the promoters to genes, and it's not just it's not just about the naming of it. If you go to tiling arrays, whole genome chip, then there's a whole issue of assigning enhancers to genes. And in Drosophila, we've got a collaborator. I didn't show any Drosophila data, but in Drosophila, our collaborator finds that about 40% of the binding events in the Drosophila genome can't be assigned to a gene with confidence because they're occurring either equidistant from distant genes, or they're occurring in a region that's far from any genes that are known. So, the chip data is quite ambiguous in terms of which genes are actually being regulated by binding events. And that's a very interesting issue.
So how do you...
Well, we were dealing with promoter arrays. Promoter arrays are simpler because they've been designed only to investigate the promoter of specific genes. So you can map the genes to the genes on the expression arrays in quite a straightforward way for promoter arrays.
Okay.
Thanks.